Hello, everyone. Happy New Year and welcome back to Science in the Age of COVID-19 in 2021. Today, we are very excited to welcome back Professor Kevan Shokat and Professor Nevan Krogan to give the very first talk in the new year. For those of you who have watched our past talks, probably remember that Dr. Shokat and Professor Shokat, pardon me, and Professor Krogan gave the third talk in this series. This was way back when we started. So today we are really very happy to have you all back with us. So before we start the talk, a very brief introduction to our speakers. Professor Kevan Shokat is a professor of cellular and molecular pharmacology at UCSF, a professor in the Department of Chemistry at UC Berkeley, and an HHMI investigator. Shokat Lab focuses on developing small molecule drugs to target a very interesting family of proteins called kinases, and they do this using a combination of synthetic organic chemistry, protein engineering, structural biology, biochemistry, and cell biology. Professor Nevan Krogan is a professor in the Department of Cellular and Molecular Pharmacology, a senior investigator at the Gladstone Institute of Data Science and Biotechnology, and the director of Quantitative Bioscience Institute, QBI, at UCSF. A bit about QBI. QBI is a collaborative group bringing together a diverse group of scientists to address biological problems in a multidisciplinary fashion. And in the wake of the COVID-19 pandemic, Professor Krogan initiated the QBI Coronavirus Research Group to form a team of national and international scientists to understand the various aspects of the COVID-19 pandemic and the therapeutic options. And today we'll hear how this international team came together to nail down the host factors involved in SARS-CoV-2 infection. So Professor Shokat, Professor Krogan, welcome back. Thank you for your time, and we look forward to your talk.
Thank you.
Thank you very much. We're delighted to come back to give you an update on our efforts on studying SARS-CoV-2 and COVID-19. And this, as you just heard, is under the umbrella of the QCRG, the QBI Coronavirus Research Group, which involves over 40 groups at QBI at UCSF, each with different expertise that we've all come together in an integrative way to try to understand this virus and ultimately contribute to identifying solutions for COVID-19. And this group got so large, we broke it into different subgroups. There's 12 different subgroups associated with QCRG. The groups are focused on different biological aspects of infection as well as different technologies as well. Here's Kevan and Brian Shokat leading the drug discovery group. And another group I just wanted to call out because I'm going to allude to it later is the structural biology subgroup led by QBI fellow Clem Verba and Professor Oren Rosenberg here at UCSF. So it's essentially hundreds of different scientists that have come together here at UCSF, at QBI, I would argue in a very unprecedented way to work together. And I'd also argue it's unprecedented the worldwide collaborations that are occurring here, not just at QCRG, but around the world. But these are our academic and industry partners that we've been working with on SARS-CoV-2 over the last 10 or 11 months. And one of the things I'm going to allude to at the end of the seminar is obviously this has been a great tragedy, but I think a silver lining in all of this has been the fact that so many scientists around the world have been working together. And I think we can all see how fast we can move when we all collaborate. So here are a couple papers that came out of QCRG. Kevan and I talked about these two papers several months ago. I'm just going to briefly touch on these. So we generated a SARS-CoV-2 human protein-protein interaction map using affinity tag purification mass spec-based approach to identify human proteins, and through drug repurposing, identified many drugs and compounds that would target the human proteins that we think the virus would need. And then we tested to see which ones had antiviral effects. So this was one aspect of studying the host to try to come up with drug repurposing. This particular paper, the other paper, we looked at the host response in a slightly different way. We looked at changes with respect to phosphorylation in infected cells in an attempt to identify the kinases that were most misregulated. And Kevan was able to identify several kinase inhibitors based on this data that did in fact have potent antiviral effects, several of which are in clinical trials right now. And just a few more words about this paper here, the protein-protein interaction analysis. So back at the end of January in 2020, I think we were the first group to clone out each one of the SARS-CoV-2 genes, approximately 30 of these genes, and we put affinity tags on each one of them, expressed them in human cells, lysed the cells, purified each one of the SARS-CoV-2 proteins, and identified the co-purifying human proteins using mass spectrometry to come up with this SARS-CoV-2 human protein-protein interaction map. And at the time that we generated these constructs, we just simply tweeted out that we had these and would be happy to share. And I just wanted to point out that we had sent these plasmids out really in a matter of weeks to, I think it's over 400 labs now in over 40 different countries. No MTAs, no strings attached, no lawyers. We just sent these out and said, please feel free to distribute. So we're happy we're able to send out these reagents to the scientific community to help in the study of SARS-CoV-2. So here was one of the initial iterations of the map that we had published. This is the SARS-CoV-2 human protein-protein interaction map. We identified over 330 human proteins. The red diamonds here are the SARS-CoV-2 proteins. The other nodes are the human proteins. And through work from Kevan and Kevan Shokat, they looked at these 332 human proteins and identified 69 different drugs and compounds that they thought would target and inhibit at least one of these human proteins. And we've been testing these drugs and compounds in collaboration with scientists in New York and in Paris, Adolfo Garcia Sastra at Mount Sinai Hospital in New York, and Marco Vignuzzi at the Pasteur Institute in Paris. And this allowed us to narrow in on a number of drugs which have antiviral effects. I want to point out that 26 of these drugs are currently being tested in clinical trials for COVID-19. This is the list of drugs that people are looking at from this particular study. I just wanted to highlight two drugs here. These are two translational inhibitors that were identified based on this map. One of them here is zotadifin. This targets a translational initiation a protein. This is a connection that came from the map. And this is a drug from the company Effector. And actually, Kevan's one of the co-founders of this company. And this is a clinical trial has been approved, and they're enrolling patients right now for this zotadifin. And then the other drug is aplidin. This actually targets a translational elongation factor, EIF1A. Initially, the connection came from Jack Taunton here at UCSF. He had identified ternatinib-4, a preclinical compound that targets EIF1A that we showed had antiviral effects. Well, it so happens there's actually a drug approved to treat multiple myeloma that targets the same protein. And we actually found that it had even a more potent antiviral effect. And I just want to tell you a little bit more about the work that's being done with aplidin. This is a paper that will hopefully be published very soon, a collaboration again with Adolfo Garcia Sastra and Chris White, who's affiliated with Adolfo. So we actually showed here in human cells that aplidin has an IC90 value that's actually a subnanomolar, which is very potent and actually 30 times more potent than remdesivir. And I should point out that Adolfo and his colleagues and our collaborators in Paris, they've screened thousands, if not 10,000 compounds and drugs for SARS-CoV-2. And this is by far the most potent one that they've seen. So this is in human cells. You can see at a much lower concentration that's was used for remdesivir in primary lung cells. You can also see potent antiviral effects. And Adolfo now has 2 mouse models where he can look to see if they have any effect on infection. One of the mouse models here is using adenovirus expressing human ACE2. They infect the mice so you could infect them with SARS-CoV-2. And then after infection, in a time-dependent manner, we're adding different drugs and then extracting the lungs from the mice to see the viral lung titers. And you can see at a much lower concentration that was used for remdesivir, you can be able to 500-fold decrease of infection using aplidin. And the company that makes aplidin is called PharmaMar, and they announced a few months ago very promising results in a phase 2 clinical trial for COVID-19. They're now starting a phase 3 trial as we speak. And Another interesting note here with aplidin, it comes from this interesting sea creature, which was found off the coast of Spain. This is where this company is, PharmaMar. And this is the compound, and there's actually a structure now. This was not from us a couple of years ago, of the compound binding to its target, EIF1A. And in this particular study, we actually, using CRISPR, make a single point mutant in EIF1A so that the compound can no longer bind. And cells that have this particular point mutant, when the virus infects these cells, they're essentially resistant to aplidin. So we've narrowed down the target that's responsible for the antiviral effects, which was a connection ultimately from our protein-protein interaction map. And we're quite excited about this particular drug, and we hope that people are going to be hearing about it in the very near future.
All right. Thanks, Nevan. I don't know if I'm switched over, Matt.
Yep, I think so.
Great. Okay, cool. I realize, Nevan, you've got the mouse, so at some point I may ask you to point at something. But well, Nevan's told you about our first drugs that we tried to identify to block viral replication. But we know that the virus does many other things to cells in order and especially to the immune system to block immune recognition. So one of the students, postdocs in my lab, Xiang Zhang, initiated an effort to understand at least one step of how SARS-CoV-2 manipulates immune evasion. So the next slide, we focused on class 1 MHC presentation because this, of course, is the system in a virally infected cell that can present virally encoded proteins, which are, of course, completely non-self and very, very well recognized by the host immune system. And if you click forward on the next panel, you see that many, many different viruses have been identified to block many steps in class 1 presentation, including the proteasome, TAP, loading, many steps. So in the next slide, Xiang asked, using the ORF collection that Nevan described in 293 T cells, if we monitor class 1 presentation on the cell surface and we express each of the viral ORFs, which one of them, or if any, control class 1 levels. And so this was an assay we repurposed from an immune oncology project we were just beginning when the pandemic started. And so the next slide shows the level of class 1 after each SARS-CoV-2 ORF is expressed. And you can see that a few of these, highlighted in red, do downregulate class 1 MHC by several fold. And in particular, NSP1, NSP14, 15, and ORF6, we decided to focus on. And you'll see that we focused most on NSP14. On the next slide, then we went back to the protein-protein interaction map and asked what was known about NSP1, 14, 15, and ORF8. And in terms of their known functions, you'll see that NSP1, 15, and ORF8 had been identified in suppressing interferon responses. That's a common feature for the cell intrinsic innate immune response. But NSP14 is a very interesting protein that has two enzymatic activities. It's a 3', 5' exonuclease and an N7 methyltransferase. So it's a fusion of two different enzyme activities, and it had no immune suppressive functions attributed to it. So we decided to focus on that. And it also had the largest magnitude of class 1 suppression. So next slide. We first confirmed that this was affecting post-transcriptionally. So now when we turn that FACS assay into a Western blot, you can see that empty vector versus NSP14 causes a decrease in class 1 MHC. And you can see that NSP14 is expressed, and this is a strep tag, but RT-PCR for the HLA components, each of the heavy chains and the beta-2 microglobulin mRNAs are not affected at all. So this is post-transcriptional. So the next slide we decided to ask which of the two enzymatic activities. Remember, I told you NSP14 has an exonuclease and a methyltransferase activity. And so we individually inactivated each of the enzyme active sites. And so the exonuclease inactive or the methyltransferase inactive. And what you can see is that if we inactivate the exonuclease, NSP14 can still downregulate class 1 MHC. But if we inactivate the methyltransferase, we see no downregulation of class 1 MHC. So the inhibitory activity comes from the methyltransferase activity. That is still a mystery, and any of you who might have an idea about how that connection could work, I'd love to hear from you. We're still investigating that. On the next slide, we asked, can we inactivate the class 1 downregulation? Of course, this is the key. Can we re-alert the immune system to a virally infected cell by inhibiting class 1 presentation suppression and then recover presentation of viral peptides to cytotoxic T cells? So IMPDH is one of the host factors we identified, inosine monophosphate dehydrogenase 2. It's involved in one of the pathways to make GTP. Mycophenolic acid is a natural product inhibitor of that enzyme. And so you see on the right, when we express NSP14, we see downregulation of class 1. But in the last lane, if we add mycophenolic acid, we rescue all the class 1 MHC. So by just inhibiting that one host factor associated with NSP14, we can recover class 1 on the cell surface. This is really exciting because mycophenolic acid is an approved drug. It's something that we could treat patients with. In fact, there are ongoing clinical trials of mycophenolic acid, not looking at this kind of immune evasion strategy, but on just primary viral replication. So again, it's exciting that all these clinical trials are going on so quickly right now that maybe we can leverage some of these findings with looking at that clinical data. The next slide shows that the key output that seems to be important for suppressing this class 1 MHC is the biochemical activity of IMPDH2. So if we inhibit IMPDH2 with mycophenolic acid, but recover GTP levels in the cell by adding exogenous guanosine, you can see in the last lane that we can resuppress class 1 MHC. So we know that the methyltransferases activity of NSP14 is important, and we know the catalytic activity of IMPDH2 and its downstream guanosine nucleotides are important for suppression. So we're clearly on target with all of these perturbations. Now, on the next slide, we wanted to know, can we actually get more peptides onto the cell surface when we give mycophenolic acid in an NSP14 expressing cell? So with empty vector, you see that when we immunoprecipitate class 1 MHC and acid elute, we can identify 200 plus peptides from human proteins. We put in NSP14, that number drops dramatically, and we get a couple of NSP14 derived peptides. But now when we add mycophenolic acid, we can get additional NSP14 peptides and additional peptides in our acid elution. So this is just from one viral protein. Now we're getting ready to do the full viral infection and look at all of the viral proteins and what their presentation would be. So finally, I think the last slide just tells you where we are. We've done this with NSP14 alone extensively. Now we're doing it in virally infected cells. And on the next bullet, we're still, as I said, stumped about how the methyltransferase is important for of NSP14 is important for class 1 MHC downregulation, and how that one factor, IMPDH2, is linked to the methyltransferase and the downregulation. So This just, I think, highlights the power of these unbiased protein-protein interaction maps that Nevan's done so much to produce and to sort of mine. But now we know these connections, we need to sort of buckle down and find out the mechanisms in the cell biology of the class 1 regulation. So I think, Nevan, you'll take it back and tell us about other SARS viruses.
Right. So thanks, Kevan. That's great. So Kevan and I are obviously discussing the science that's been extracted from the SARS-CoV-2 human interaction map, but I think there's great value in looking at some of these other coronaviruses that have wreaked havoc since the turn of the century, not as problematic as SARS-CoV-2, but they have been problematic, such as SARS-CoV-1 in 2002 and MERS in 2012. And one of the logics here is, can we look back at these other coronaviruses and compare it to SARS-CoV-2 to be in a better predictive situation for the next pandemic, the SARS-CoV-3, because we all ultimately know that it's coming. And if we can focus our attention on identifying a host-directed therapy that would be working for SARS-CoV-2 and SARS-CoV-1 and MERS, almost certainly is going to be working for SARS-CoV-3. So that's a logic at looking at a number of these different coronaviruses. And this work was recently published in print a few weeks ago, this comparative host coronavirus protein interaction network revealing pan-viral disease mechanism with the ultimate goal of identifying, as I said, pan-viral therapeutic strategies. And I just wanted to point out here that there's 200 authors on this paper from 14 different institutions in 6 different countries, a truly worldwide collaborative effort. And because we work together, it was the reason we could move so fast on this multidisciplinary study. Here is an overview of this study. So as I said, we didn't just look at SARS-CoV-2, we looked at SARS-CoV-1 and MERS. We cloned out each one of the genes, just like we did for SARS-CoV-2, to generate protein-protein interaction maps using the same strategy that I described for SARS-CoV-2 to generate these maps. And in parallel, we also carried out microscopy experiments to localize in which subcellular compartment each one of the individual proteins was residing in. We also created 14 different antibodies for 14 different SARS-CoV-2 proteins, so we could do this localization in infected cells as well. We merged all this information together, combined it with functional data, CRISPR and RNAi based genetic analysis of the host factors. This allowed us to pinpoint specific interactions and proteins that we wanted to structurally characterize using primarily cryo-EM. And we also merged all this information with real-world clinical data as well to see if some of our drug predictions actually had a value. So here is the SARS-CoV-1 human protein-protein interaction map that we had generated, identified over 350 human proteins here corresponding to 25 different SARS-CoV-2 proteins. Here's the MERS human interaction map that was generated. So 22 MERS-CoV proteins corresponding to almost 300 human proteins. And there's a number of different ways you can compare these 3 different maps. I think I just like simply hierarchical clustering, simple but quite powerful. And it allowed us to identify these different groups. And for me, what I was most excited about were these 3 groups. The first one here is about a fifth of the interactions you can see are shared across all 3 viruses. About a quarter of the interactions are exclusive to SARS-1 and SARS-2, and about a quarter of them are MERS specific. And we worked with Trey Eidecker at UCSD on a differential scoring system. This is something we've developed with him to look at cancer networks across disease and cancerous cells. I won't go into the math of this, but I'll just say that we use this to highlight these 3 different categories that we were most interested in. And on this particular network, the edges represent the 3 different categories. So if an edge is black, it means we found an interaction with all 3 viruses. If it's blue, we found it more MERS specific. And if it's red, it's more SARS-1 and SARS-2 specific. So there's a wealth of biology represented on this particular map. It contains all 3 maps together. And just to zoom in on a couple of connections, just to show you the value of this cross comparison. So we found that NSP6 only with SARS-1 and SARS-2 and not MERS physically interacts with sigma R1. We've been focused on a lot of sigma R1 modulators over the last year. So we only see this with SARS-1 and SARS-2 and not MERS. Here's the end protein. We find a number of black edges here, meaning we find these connections in all three viruses, including a number connected to RNA processing, as well as a connection here to casein kinase 2. This was a kinase that came out of our global phosphorylation work, and this led us to a casein kinase 2 inhibitor showing antiviral effects, and this is a drug presently in clinical trials right now. And the idea would be, the hypothesis would be that this drug would be hitting all three of these viruses and other corner viruses as well, and we're looking into that. And then the last connection is a NSP14, which Kevan talked about. There's a number of blue edges here saying that these are MERS-specific interactions. We don't see them with SARS-1 and SARS-2, with factors linked to DNA damage repair and response and cell cycle arrest. Very well-studied DNA damage proteins, including the MRX complex, MR11, RAD50, and then P53, probably the most well-characterized cancer protein. So I think it's very interesting why NSP14 only in MERS would be interacting with these DNA repair damage factors, and look into more detail into that. And I should say here, although I don't have it on this slide, NSP14 has a black edge to IMPDH2, which is the factor that Kevan just told you about. So the phenomenon that he's talking about almost certainly would be conserved across these different corner viruses. And in data, I know that Kevan didn't show you, he has preliminary information suggesting that is indeed the case. This is a very powerful way, I think, to look at these different maps by merging them together and doing these differential interaction scores. So now a few words about some of the localization work that we did. So as I said, we did transient transfections of each one of these proteins into human cells, and then did microscopy. This was work led by Andrew Pedden at Sheffield in England and Robert Gross at the University of Freiburg in Germany. And as I said before, in collaboration with Massimo Palmarini in Glasgow, he had generated 14 different antibodies against 14 different SARS-CoV-2 proteins so that we could do the localization in infected cells as well. And Svenja and Robert's lab was spearheading a lot of this effort, and we do all of our work, as I've been alluding to, with Adolfo Garcia Sastra in New York. So we generated a lot of images in collaboration with this great group, and all of this data is available, obviously, to the scientific community. We tried to summarize the localization patterns here. On the left, this is data, again, just from the transient transfection experiments, where the more blue the box is, the more we think that it's localized to that particular compartment. And we overlaid on top the localization patterns that we deduced from infected cells using the endogenous antibodies. And this is using these three different colored boxes. So it's overlaid here with SARS-CoV-2. So there's some differences, there's some similarities, but I think this is a great summary of the localization of all of these proteins. I want to highlight one particular protein. This is ORF9b that you see in SARS-CoV-2 and SARS-CoV-1. It doesn't exist in MERS. And you can see it's the only protein in both of these viruses that localizes to the mitochondria. And you can see that when you do transient transfection, and you can also see this when you look using the endogenous antibody in infected cells. And this is the staining pattern from that particular experiment. This, I would argue, is a quintessential mitochondrial stain that you can see that ORF9b is localizing to. So I want you to remember that ORF9b localizes to the mitochondria, because I'm going to be coming back to that connection in just a few minutes. So as I said before, in parallel, we're also doing genetic validation of these protein-protein interactions, and we're doing this in two ways. Collaborating with Marco and Veronica at the Pasteur Institute, they're using RNAi in A549 cells expressing ACE2, individually knocking down each one of these factors and seeing what effects they have on infection using RT-qPCR. And in parallel, we collaborated with a company, Synthago, in South San Francisco. These are some two great young scientists here, Jared and Kevin, where they knocked out each one of our genes one at a time using CRISPR in CACO2 cells. And we shipped those cells to Chris Bassler at Atlanta at Georgia State University. And he looked to see what effects these knockouts had on infection using a different assay. He was taking the virus off cells and then reinfecting other cells, and then doing using an anti-N antibody for readout of infection. So we were looking at two different cell types, two different genetic perturbation strategies, two different assays in two different continents to give us a summary of the genes that we found, at least in one of these systems, had an impact on infection when removed or depleted. So we got 73 in total. Here's a scatter plot of what we found. So the nodes in red are genes that, when knocked down in A549 cells, have an effect on infection, and the ones in yellow are from CACPA2 cells. The one in orange are ones that we see in both. And I just want to point out here, sigma-1 is actually a gene that we found in both strategies as having an impact on infection. So again, there's some similarities, there's some differences, but overall, we identified out of the 332, 73 different genes. And as I'm sure many people are aware of on this call, there's been a series of CRISPR screens that have been published over the last few weeks. 5 actually, one that we're involved in here, led by Andreas Pushnik. And I just want to point out this one too from Charlie Rice. They actually targeted our 332 genes in this particular paper, and they actually found a very good overlap between what we found and what they found. But overall, it's very exciting to have all this genetic data, and we're looking at it as are others, integrating it together and looking at it, the overlap, not just with respect to individual genes, but protein complexes and pathways where you see a greater overlap. And we're also combining it with all the other data that we're generating as well. So it's great to see so many scientists generating complementary data that the community can ultimately be using. But here's a summary of our genetic data from these 2 different cell types. Here, these blue nodes are represent the 73 genes. And then I'm highlighting if we found a physical connection with proteins from SARS-CoV-1 in orange, MERS in yellow, or SARS-CoV-2 in red. And I want to just zoom up on one particular connection here. It's ORF9b again. Remember, I told you it localized to the mitochondria. And we found it physically associating with TOM70, a mitochondrial protein. And when you knock down TOM70, it has a statistically significant impact on coronavirus infection. And we've been looking at these interactions structurally as well. This is one that had multiple lines of evidence connecting it to SARS-CoV-2 infection, this TOM70. And this is where this QCRG structural biology subgroup has been so important. This is led, as I said, by Oren and Klim, involving 60 different researchers, mostly trainees, mostly students and postdocs from 18 different labs. So they've been marching along and characterizing structurally, not just the SARS-CoV-2 proteins, but the interacting proteins as well. So they generated this cryo-EM structure of TOM70 binding to ORF9b. It's a 3 angstrom structure. And you can see ORF9b is tucking itself up here into the active site of TOM70. So what is TOM70? It's actually a chaperone that shuttles proteins in and out of the mitochondria. So the question is, what is ORF9b doing with TOM70? So maybe it's using it to get itself in the mitochondria, or maybe it's altering the shuttling of other host proteins that are ultimately beneficial for viral infection. And this is something that we're looking into in greater detail to see what is the underlying mechanism. A few more comments about the structure. The interaction is mostly hydrophobic with 36 out of the 38 residues interacting with the TOM70. And there's 2 phosphorylation sites that we've identified in our phosphorylation paper on S50 and S53 on ORF9b. And they're actually tucked up in here in the active site. And based on the model here, if these residues are phosphorylated, there's no way that ORF9b could be interacting with TOM70. And data I'm not going to show you from a postdoc, Jody Batra, she actually made phosphomimic mutants. And indeed, that theory was true, that when you mutate these residues in that way, you no longer see ORF9b associated with TOM70. We think we know the kinase too, based on our protein-protein interaction work. Just another comment here also about the structure of ORF9b. It's very interesting. We call it the shapeshifter, because when ORF9b is by itself, it forms a dimer like this, and you see these beta sheets predominantly in the structure. But when it's by itself, it forms a very different structure. You have this alpha helices, and that's the form that actually binds to ORF9b. And this is one of the theories we have with coronavirus proteins and maybe viral proteins in general. Maybe many of them have these different configurations. That's how they can have a bigger impact on the host cell with such a limited genomic capacity. So we're looking at this phenomenon in more detail, not just with SARS-CoV-2, but with other viruses as well. And I really like this example too, because it's an example of how you combine the proteomics, the cell biology, the microscopy, the genetics, the biochemistry, and the structural biology to narrow in on this particular connection, which, by the way, is conserved in SARS-CoV-1. And right now, in collaboration with Kevan Shokat, we're looking at this structure, and Brian's making predictions about compounds based on the structure that could be inhibiting, if not the interaction, the function of this particular protein-protein interaction. So hopefully this particular connection will have some value in the future for this pandemic and maybe for the next one as well. And I just want to end by tell you about some other structural work that's been ongoing. I think, as everyone's aware, last year, Google and DeepMind and AlphaFold made a huge splash showing how they could use AI to really predict the structure of proteins. And what they had done is pick 5 proteins and just put it on their website randomly. And one of them, ironically, was NSP2. And they tried to predict the structure of NSP2 from SARS-CoV-2. And when you looked at the structure, the domains were actually probably right, but the overall structure the configuration was all wrong. Well, Klim and his team actually had a cryo-EM structure of NSP2, but not the full structure. It was about 70% the N-terminal domain. The last little bit, it was too fuzzy for them. But what Klim did was notice this was on the website, and he took this prediction from AlphaFold with his cryo-EM structure and ultimately elucidated, which was an accurate structure of NSP2. So this last little bit actually came from the AI. And I would argue this is probably the way of the future, where you're going to be combining cryo-EM, maybe with tomography too, with AI. So cryo-EM will get you part of the way it's there, and then AI can maybe fill this in. So we're very excited about what's happening here in the world of structural biology and combining it with AI. And I should also point out, if you go back and look at our interaction map, there's a number of interesting interactions, including this one with this protein RAP1 GDS1, and Klim and Oren and their team have a cryo-EM structure of NSP2 binding to RAP1 GDS1. It's a lower resolution. In pink here is NSP2, and in blue we have RAP1 GDS1. But the hope would be that they're going to have a higher resolution of this particular interaction. So these guys have done a remarkable job marching through these interactions, and they're characterizing them structurally with this team that they've put in place. And the last thing I just want to say about NSP2, we've been looking at the structure and looking for variants that exist in the wild, if you will, that fall on the surface of NSP2. We've identified one that's particularly prevalent. It's about in 20% of the viruses that are out there. It's more prevalent in South America and in Europe. And in collaboration with Adolfo Garcia-Sastre, we've shown that particular variant actually has a phenotype in the lab. And now we're seeing if this particular variant has an effect on protein-protein interactions. And just a few words more along those lines, the fact that we're setting up this infrastructure to ultimately study these variants. Everyone's familiar with the UK variant. It has 23 different mutations associated with them, 17 of which result in changes of the proteins. 8 of those are in the spike. So presumably that's what's contributing a lot to this increased transmissibility. We're studying these a variety of different ways. We're also looking at these other mutations and seeing what effects they have on the map. There's 3 in NSP3. There's one interesting 2 amino acid deletion in NSP6. There's an ORF8 deletion that Kevan talked about, ORF8, which we're really interested in. And then there's a couple of mutations here in N as well. So we're studying all of these mutations systematically using the tools that we've described. And the last thing I just say is I think this is the importance, I think, of studying the host and going after host-directed therapies. Because if a treatment will have an impact on SARS-CoV-2 and SARS-CoV-1, there's no way SARS-CoV-2 will be able to mutate away from the reliance on that particular host factor. So these vaccines, it looks like they're going to be working for this UK variant, hopefully for other variants in the future, but I think we're going to need more tools here to fight this virus. And I think doing the host-directed therapies using the type of information that Kevan and I are talking about to lead the way, I think is going to be very important going forward, not just for this pandemic, but for the next pandemic as well. And I just want to segue here to the last point here that I'd like to make. And the thing I'd like to say at the end here is there's a lot about the scientific enterprise I don't agree with. And I think the problem here, one of the major problems in our business is that science is incredibly siloed. One person gets the award, one person gets the grant, one person gets tenure. And oftentimes I would argue that the system actually discourages from collaboration. But during this pandemic, you can see how exciting the science can be and how fast science can move when you break down these silos, and you break down the silos across different laboratories, across different institutions, and between the academic world and pharmaceutical companies. So the challenge, I think, for us is to try to keep this infrastructure and this spirit in place so we're better prepared for the next pandemic. And I would say, why aren't we working on all diseases like this? Why aren't we working on breast cancer and Alzheimer's and Parkinson's? So I would challenge all of us to keep in this collaborative mode as we attack other diseases going forward. So with that, we'd like to thank you for your attention, and we'd be happy to take any questions.
Nevan, you forgot to thank another person in the audience.
Oh, sorry. Yeah, I got, thanks, Kevan. I got, my mom is watching. So hi, Junie, and my sister Lana as well. So I hope they stayed on till the end.
All right, you guys, that was just completely amazing again. Great science. Yeah, great rant. I agree. Burn down the existing scientific culture and rebuild. And yeah, let's deal with problems before, it's way too late. Gosh, okay, I've got a billion questions. A reminder, audience, please go ahead and either type your question into the box and or raise your hand, and we will keep an eye on those. I'll get things going. So you guys looked at the protein localization in, I don't know these lines personally, A549 and CACO2, but have you looked at localization in various primary cell cultures like pneumocytes or something like that? Because, cultured cells, can be a bit dodgy in terms of what you think you see. So
yeah, I mean so clearly using, that's a great point, a great question. Using the antibodies in doing this in infected cells is much more tractable going across different cell types, and that's exactly what's happening. These antibodies are being used. They're actually being used also to pull down proteins in infected cells, so we can see how the interactions change. But as you point out, that we and others are looking across different cell types, and in particular primary cells, which would be very exciting, as you point out, to see what the localization patterns are as well. And you can do that in a temporal way as well. So that is work that's ongoing, and that would be particularly pertinent with these 14 different antibodies. And there's efforts now ongoing to try to get antibodies for the rest of the SARS-CoV-2 proteins and the other coronavirus proteins as well.
Also, add one quick thing, Lauren, on localization, but as we repurposed cancer drugs into these host factor targeting, the sort of therapeutic window in those A549 and the CACOs, which are transformed cells, were often much narrower than when we tested them in primary bronchial cells with viral infection. Then the tolerability increased. So your point on the therapeutic side is really, really important also.
Yeah, I love that set of studies of looking at localization, and then you're right, just follow your nose at that point, do pull downs, etc. So follow up. Why do you think the subcellular localization is so different between the coronaviruses? It was pretty similar between SARS-1 and SARS-2, but then you look at MERS and it's like, half the proteins are going to, different organelles, which is, viruses, they just do whatever they can. But I thought that was pretty shocking, that they would change localization even.
And Pedro Beltrao, who is at EBI-EMBL, who's been working closely with us, is a great computational biologist. He has been comparing the differences and similarities, but the differences in localization to the differences of protein-protein interactions, and also the conservation of the individual proteins. And he's seen some very interesting trends. It's like, all right, the more conserved these proteins are across coronaviruses, the more they have similar interactions, and the more they have similar localization patterns. But then the opposite is true as well. So I think there's just some really cool biology to be had. Obviously what Kevan and I are pushing is kind of a pan-coronaviral approach. Let's look where the commonalities are and, use this information to come up with a strategy that could target these and the next one. But the differences are, from a scientific point of view, are just as interesting. And we're going deep in not just the commonalities, but the differences as you're pointing out. So there's a ton of biology here to be had. in general. And if you just want to understand important mammalian molecular biology, viruses are a great tool.
Yeah.
They tell you what to work on.
Yeah.
They're smarter than us. So they're like, all right, just let's generate the data and tell us what to work on. And that's what viruses do.
Yeah. Okay, I'm going to ask one more and then pass it over to Sarada. Yeah, I mean, super exciting preliminary results with Aplidin and Zotadifin. I'll ask the obvious question. You know, how would EF1 alpha inhibitors, you know, isn't that going to have a billion side effects? I mean, that's kind of it's a pretty big sledgehammer.
Kevan, do you want to answer?
I mean, you know, what I think people's intuition is that once you have an mRNA, you've got the protein. But of course, translational regulation is very, very sophisticated. So a lot of, you know, more of these onco-mRNAs or the mRNAs probably that the virus has, which needs to be highly translated, rely on this sort of high capacity translational machinery. So it's not that you're blocking it 100%. That's what's great about the pharmacology. You're sort of dampening it down and then chopping off the top end. And that's what we think both with Zotadifin and Aplidin. Zotadifin, 4A inhibitor, not all mRNAs rely on 4A. So there's that aspect. I mean, but it's a great question. We wouldn't have thought there would be such a big therapeutic index.
But what I'd add too, in terms of the toxicity, I mean, I forgot to mention that Kevan's drug, I call it Zotadifin, he calls it Zotadifin. It's the same drug. That and Aplidin are being used to treat multiple myeloma, and that's being used to treat for, you know, relatively long periods of time. If you're just going to introduce this to someone who's got the virus for a week, that's, you know, you don't have to worry so much about side effects and toxicity if it's a week versus years, right? So that's another
excellent point. Yeah, that's
very fair. Very fair. Yeah, if this will get you out of the woods, you know, over a week course, totally. Sarada, you want to go now? And audience, come on, step up, come on, guys. There's so much here. Ask questions.
Do I call on my mom to ask a question here?
So, following up on the comparative study, I'm still looking at the data set. So a quick look, MERS clearly appears to be more distinct than SARS 1 and 2, but even looking at your data set, are you able to predict why SARS 2 is different from SARS 1? For example, the transmission appears to be different, and in general, SARS 2 is being sort of difficult than SARS 1.
Yeah, I mean, it's a great question. You know, obviously we put out all the data for everybody to look at. In the initial publication, we decided to kind of lump SARS-1 and SARS-2 together, because as you point out, they're much more similar than MERS, to kind of accentuate the differences. But right now, we're doing exactly what you're saying, is we're going deeper into the differences of SARS-1 and SARS-2, and we're trying to make predictions about, you know, why one is more lethal versus why the other one's more transmissible at a molecular level. So there's a number of interesting connections that are there that I hope many others are looking at as well, that we're trying to kind of connect to these differences that you see in people. So it's a great point, a great question, and we're focused on that right now.
Oh, thank you. And speaking of predictions, I mean, at this moment, the UK variant or the new variants is probably in all our minds at the moment. So I know maybe I missed this in your slide, but are you even able to predict based on your data set and your interaction idea? Do you think the UK, the new variants, would they behave any different from the original strain that's been floating around?
Well, we're collaborating with the lab in London right now, the Towers lab that has the virus in the lab, and it looks like they are seeing differences in the laboratory setting. We're going to be doing similar things that we've done before in terms of phosphorylation analysis. We're doing that right now. And Kevan can then come in with his kinase inhibitors and see, you know, what's different or what's the same. I think overall, as we were kind of talking about, I think the drugs that are hitting the host factors that have an effect on the older variant will almost certainly have an effect on the new variant. But you know, we have to collect the data on that. But I suspect a lot of the molecular data would be similar, but kind of like we talked about with SARS-1 and SARS-2, a lot of the interest will be in what's different.
Mhm.
So the idea is to kind of run the whole gamut on this new variant using all the tools that we described and look for what is ultimately different. I think a lot of it is going to be attributable to the spike mutants,
Mhm.
but I think there's other mutations in there that could be contributing as well. So it's scary, but it's also scientifically exciting to be looking at this particular virus.
Right. Thanks.
Sarada, you want to ask our
You go ahead. It's an audience question, Lauren. Why don't you ask that?
well, yeah. So Arad, can you unmute yourself and we will call on you? Okay.
So if Arad can't, I can state the question.
Here he comes. Here he comes.
Oh, good. Perfect.
Can you hear me?
Yes.
Yeah.
So I believe the first plot that Dr. Shokat showed with the MPA treatment, it looked like the protein levels of NSP14 also kind of went down. I'm curious if you think that maybe IMPDH2 might potentially be regulating the stability of NSP14 itself.
Yeah, great catch. That is what we think. I don't know if, anyways, yeah, it's pretty clear, like you saw from that slide 16, that when we add mycophenolic acid, the NSP14 drops. And you might be familiar, IMPDH has this very filamentous structure depending on the guanosine, the GTP levels in the cell. So that is one thing we're definitely thinking, that maybe NSP14 associates with IMPDH and is stabilized by that interaction. And that's how and then it also participates. And one neat thing is if IMPDH makes guanosine, then that's it getting methylated by the N7 side. So it's that physical interaction is something more important than for stability too. Great catch.
Thank you.
That's awesome. Okay, audience, ask more questions. Until then, let's try something a little different. So let's try to brainstorm real fast and help you guys. So this absolutely mind-blowing result about that the effect of NSP14 on MHC expression levels is coming from the RNA methyltransferase activity. So do you guys have, I mean, you said you didn't know what was going on. Do you have an inkling of what's going on? Like, so I'll just toss out, I'll toss out a straw man. So do you think that it is, okay, first straw man. Do you think it is that precise enzymatic activity that's doing it, or do those enzymes have side reactions that might be doing the thing?
Yeah, like a neomorphic function that is somehow. One thing I don't think we showed is that, connected to sort of Arad's point, is that when we make the point mutation in the methyltransferase, it seems to lessen the interaction with NSP14. And so we think that also leads to destabilization. So it could be that sort of indirect thing again. And we could have been a little bit fooled, something about enzymatic activity and structure. Yeah, that's a wrinkle.
Oh, so how about maybe making some other mutations that aren't in the active site, but are in the protein-protein interaction?
Yeah, that's what Clem and the structural biology group is trying to solve, the IMPDH2 structure with NSP14. Actually, I was messaging him on Slack when Nevan was talking, asking where we were on that. And he said they've been making IMPDH mutants that have more stable structures to get the structure of the 14, so we could make those interaction mutants. Yeah. Nevan's group's got some really great sort of structure prediction ways to figure out what are the two surfaces that are interacting. It might be good to apply it there.
Yeah. Okay, but so for part 2, I'll make the counterpoint. So I was looking, I just pulled up, some articles on N7 RNA methyltransferases. And it seems like one of their favorite targets are mRNA end caps. And so one could imagine that, some charge on steric alterations there would affect docking to the ribosome. You've shown that it's post-transcriptional, but it could be translational regulation. So,
Wow.
how about maybe doing some in vitro translation assays?
And just spike in.
Mm-hmm.
Wow, that's a good suggestion. We have those assays going for a different project. So we could just put in NSP14 and the different mutants and see on a vanilla mRNA. I like that.
I guess also in the spirit of Lauren's brainstorming here, I was just thinking about one of the 14 antibodies, Kevan, is one of them is against NSP14, and we're working with a collaborator in San Antonio that can genetically manipulate the virus, and we're working with them to put in that mutation into the virus, Kevan's mutant, and then what we could do is pull down in infected cells with the antibody, the wild type and the mutant, and just to see, just to get a little bit more insight into maybe what that mutant is doing. So we could talk to Lorraine and we should set that up, Kevan.
Yeah, I think we're trying to get that meeting set up right now.
So the obvious question would be, how do you think this is specific to MHC class 1? And if so, how could something like this plausibly be restricted to, that small a subset of genes?
Yeah, no, that's a good question. I mean, I didn't show, we checked other cell surface things, and they're not downregulated. So it's, to the extent that we checked a few things, it's not just globally sort of shaving off membrane protein integration. But that is, again, that's what the power of the virus is. So now the question is, we've got to design a better experiment.
Have you dug any deeper? Is it like, do you know, is it A, B, or C, or?
Yeah, I forget. In fact, I think we checked, and I think it's all three, but HIV does something interesting, which is when it knocks the HLAs down, it knocks down HLA A and B, but leaves C up so that natural killer cells don't kill it. And C is the least highly expressed on the cell surface. So HIV selectively sort of left the one up to prevent natural killer cells, but downregulated the most of expression that would present viral. So I think, I actually can't remember if we did that experiment. It's another good one.
Yeah, and I think there are T cell lines and NK cell lines that express, preferentially express different subunit combinations. So.
Yeah, we've got some that are null and we can express whatever we want and things like that. T1 cells, T2, I forget.
This is just so mysterious.
Okay, I have a question here. So going back to your protein phosphorylation study, it appeared like casein kinase 3 appeared to be playing a role. Do you have an idea as to what the mechanism could be?
Casein kinase 2. So there was, I guess, multiple data suggesting casein kinase 2 is connected. One is the fact that the N protein physically associates with casein kinase 2. That's number one. Number two, if you look globally at our phosphorylation studies and map back to the kinases that are responsible, the most upregulated in infected cells was casein kinase 2. And then the drug that is in clinical trial right now for COVID that targets casein kinase 2, I can't pronounce the name, but it had a strong antiviral effect. And I guess the last piece of data I'll say, and we didn't mention this or we didn't talk about this, but with Robert Gross at the University of Freiburg, he had actually showed that in infected cells, Philippodia get formed. These are these kind of stringy-like protrusions that that come out from infected cells and poke holes in other cells, and it's a way for the virus in an ACE2 independent manner to infect other cells. And what he showed through microscopy is that actually casein kinase 2 localizes to this filopodia. And it's known that casein kinase 2 is used to manipulate the actin cytoskeleton. So our theory here is that the virus is using casein kinase 2 to help make this filopodia, and which will help infect other cells. And we're testing that right now collaboratively with Robert Gross, and actually Doug Mullins as well here at UCSF. So casein kinase 2 does a lot of things, and the virus is probably manipulating it for multiple things, but this is one thing we think that the virus is using it for, and we're looking in more detail in that connection using a multitude of different approaches.
Yeah. What do you think the virus is doing messing with the mitochondria?
Well, that's the powerhouse of the cell, right? So that's a good switchboard to be in control of, right? So
well, but what would a virus want to do with the mito?
Probably manipulate the cell in a way where it keeps it alive long enough so that it can replicate enough, or it bursts and kills, right? So it's probably manipulated it in a way so that the cell doesn't want to kill itself, I suspect.
Oh, right. Yeah, that is okay. So shut off apoptosis before it gets a chance. Oh, okay.
That's a theory, but
yeah. Okay.
But when you look at all these interactions, the virus gets his fingers in pretty much every major biological process and every major organelle, which is both exciting and scary, I guess, at the same time. And we generated a lot of these different maps across different viruses. I've never seen a virus like SARS-CoV-2 get connected to so many different pathways and different processes and different organelles. Maybe this is one of the reasons, at least at a molecular level, why it's been so problematic to fight.
Yeah.
I think one cool thing about that, if it puts its fingers in so many places, one thing we did on the phosphorylation is the time course. And I think there's still a lot to sort of mine from that, because if you're going to mess with so many things, you usually do it in a very ordered way. And that could give us a lot of insight, I think.
And, we've also now had data, which we're looking at right now, is ubiquitination, another PTM. Phospho and UB often go hand in hand, but UB gets you at different areas of biology. So we're doing those comparisons. And then, as I said, looking at the UK variant, doing the exact same thing, comparing the phospho and the UB across the old and the new SARS-CoV-2 variant. So hopefully that'll be revealing, as Kevan is mentioning.
I mean, do you expect that would be different?
Well, I would expect it to be, for the most part, similar. And the question would be we're looking at these mutations associated with the variant. We're doing protein-protein interactions.
Yeah.
We're going to be looking at localization. We're going to be looking at phospho. We're going to be looking at genetics. So, if you see a difference of a protein-protein interaction, can that then be, made sense of with respect to the phospho with the genetics and the pharmacology and everything. So we're throwing the kitchen sink at it. And then the idea would be, okay, where there is differences, hopefully multiple different data types point in the same direction in terms of where it's different. And so that's the goal. That's the power of these multi-pronged approaches. I suspect overall it's going to be very, very similar, which I think is a good thing, because that means if you have a host-directed therapy, that'll be working against that. But where there are differences, and there will be some, I think that'll be very interesting as well. Maybe that information could be used to predict the next virus that's going to undergo zoonosis.
So this, again, going back to comparative study, do SARS-1 and MERS also form filopodia?
It's a good question. I don't think that's been. I suspect it would. I don't think it's been looked at. To my knowledge, that has not been looked at. But if casein kinase 2 is involved, we found N protein interact with casein kinase 2 across all three viruses. The theory would be, if that's a key connection, then filopodia would be formed. But there's other viruses that form filopodia. Influenza does it at a much lower rate, and vaccina does it as well. But to my knowledge, when I talk to the virologists, they say they've never seen this level of filopodia formation before with any other virus. And you get these branching of the filopodia, which makes it even creepier, right? So that you can be poking more holes in the adjacent cell, increasing infectivity rates. So to my knowledge, it's never been to this level of filopodia formation along with the branching that occurs with the filopodia, which is kind of cool, but also scary at the same time.
Yes.
I'm just thinking of ACE2 independent transmission. So, and again, SARS-2 is so highly transmissible. So connecting the dots, maybe if this filopodia thing is specific to SARS-2, or if it's even enhanced in SARS-2, maybe that could be the reason one of the reasons for the enhanced transmission.
Yeah, the increased, I mean, that's a very interesting hypothesis. Is that one theory could be, well, SARS-1 doesn't transmit as well, seemingly as SARS-2, and maybe that the filopodia formation isn't as prevalent in SARS-1 or even exist at all. That's a great theory, actually. So we should look into that. That's great.
Yeah, can you inhibit filopodia formation in culture?
Well, we're trying to do it with the casein kinase 2 inhibitor to see if that would actually inhibit the filopodia formation. And Robert Gross in Freiburg is looking at that right now.
Oh, guys, Lauren and Sarada, I've got a hard stop. I got to jump onto another call. But this was great, as always. Thanks for the suggestions. I'm madly taking notes on those. Thanks for the brainstorming.
Yeah, very fruitful, very fruitful.
Yeah.
Hey, just before you go, one question to leave you with. Do these filopodia contain extracellular matrix creating proteases?
That's a great question. Don't know. It'd be great if you could slice off those tentacles and mass spec on them, but I think that's
that looks like something Dyke. I bet Dyke has a little instrument for that.
I'm sure Dyke could figure out how to do that. We should talk to him about that. But that's a great question. I don't know. I don't think anyone knows.
Okay, well, guys, I mean, this was just glorious. So thanks as always.
Appreciate it.
Thanks, everybody.
Stay well, everybody.
All right.
Thank you.